Hello, Jenny.
I see Yangxing.
Hi, Jenny. How are you?
How are you? Good to see you.
Nice to see you.
Yeah, are you in Dallas or?
No, I'm in Tsinghua.
Oh, that's right.
Okay, so
Oh.
so let's get started. So welcome everyone to today's Inflammasome seminar. It's really my great pleasure to welcome today's speaker, Dr. Jenny Ting from from University of North Carolina. So just want to give a very brief introduction about Jenny. So Jenny is currently a William Professor of Genetics in University of North Carolina at Chapel Hill. And Jenny got her PhD in microbiology and immunology from Northwestern University, Chicago. And then she she did her postdoc in Department of Microbiology and Immunology in University of South California Medical Center, and then followed with a short period of a postdoc in Department of Microbiology and Immunology in Duke. So, and then she started her independent group from University of North Carolina as an assistant professor. And then to 2008, she became a founding co-director in Inflammatory Disease Institution, also founding director of Center for Translational Immunology. And 2009, Jenny is become William Rand Keenan Professor of Microbiology and Immunology, also Professor of Genetics in UNC. So Jenny's lab has been studying the transcription master regulator of MHC class 2 gene, which called C2TA, which is a class 2 transactivator for a lot of years. And based on the structure of this molecule, Jenny found a large family of genes that encode similar structure motifs as C2TA. This family was named the NLR. I think a lot of people actually working on this and very familiar with this. She is really a founder and the pioneer of the innate immunity. And these molecules later proved are important for immune defense against bacterials, virus, fungi, parasites, and damage-associated molecules. So also, Jenny's lab studied the biological effect of these NLR proteins, which in different disease models. But in addition to these, her lab also studying different projects involving the microbiome neural inflammation and also immune activating microparticle delivery system. So Jenny is interesting also has a lot of implication in different human diseases, including multiple sclerosis, cancer, autoimmune diseases, biological therapy, infection, and inflammation. I think a lot of us actually have been studying and learned her work from her literature, published paper. So just a few words about Jenny's achievement. She was awarded as Hyman Battle Distinguished Cancer Research Award from 2017. American Associate of Immunologists, AI, is a president from 2020 to 2021. International Cytokine and Interference Society Pfizer Award from 2021, and the Mentor Award for Lifetime Achievement from University of North Carolina on 2022. So I think without further ado, we are really excited to have you, Jenny. Please take away.
Thank you so much, John, for such a generous and kind introduction. I apologize because all of a sudden my computer refused to charge. So I only have an hour left. So I had to send Chuan my talk. So he has to advance the talks and I won't be able to point to things. But next slide, please. So as Chuan was saying, there are many different innate immune receptors, and this is just an old slide I've used for years, indicating that there are surface molecules such as toll-like receptors, C-type lectin receptors. But when we think about innate immune receptor, one was really novel feature is that they're actually inside the cell. So they're inside the cell to detect lots of perturbance. They're inside the cell to sense pathogens and also to sense changes in the environment. And so the genes that I will be really focused on include these NLR proteins, and the official name is really nucleotide binding, leucine-rich repeat containing proteins, but they're also known as NOD-like receptors. And then I'll talk on this protein as well in red called AIM2. And next slide, please. So as introduced, we started this whole area because we were looking at C2TA. So the NLR proteins have lots of different functions, and the major group of function is really on inflammasome, and that's shown in the second part on the left right-hand side. So there's a number of NLRs that have been shown to mediate inflammasome functions. On the top is the C2TA, which we felt is the founding member of this family. Of course, below this, you have the NOD1, NOD2, which regulates NF-kappa B map kinases. And there are a number of proteins that we have found, and many people actually around, and especially in China, have found many of these proteins as regulators of NF-kappa B map kinases, and many times as negative regulators, which is exactly what we have been showing. Next slide, please. So what I like to do is really give you a very broad view of what these proteins do in terms of NLRs, how broad the functions are. And this is just one of the many innate immune receptors. I'm sure all of you have heard of the inflammasome or know quite a bit about it. And again, many of the wonderful work of inflammasome is done at both NIH as well as many labs in China. But there are a number of inflammasome receptors or sensors. So if we know that it binds to something, it's called a receptor. If we don't know what it's binding to, then we call it a sensor. So on the left-hand side, you have NLRP1. That is a pretty well-known NLR, the first one that defined the inflammasome. And it is activated in human by DDP8 or 9 inhibition. And mice is activated by the same inhibitor, but also by lethal factor from anthrax. And on the other side, you also have other inflammasomes, and I just have it in red box as AIM2. And this is a very well-known inflammasome protein, and that recognizes DNA and was found by 3 different groups. Second to your left is the NLRP3 inflammasome. And I just will go through this, and this was found to both conduct a canonical inflammasome function or non-canonical. In the canonical function, you have the caspase 1, non-canonical is caspase 11, and what is known is that a common adapter called ASC, indicated here by the blue box, is a common adapter among many inflammasome proteins. And so NLRP3 and ASC and caspase-1 can form this large complex that, when associated, causes the autocatalytic cleavage of procaspase-1 into caspase-1. And then it activates pro-IL-1 and pro-IL-18 down here at the bottom by cleaving these pro-proteins into their mature form, or cleaving gasdermin D into their mature form. And so this is the process of the inflammasome. And next slide, please. And I won't go into the details, but just the things that we have found. And many times when we found these things, there's a paper published side by side at the same time that indicated inflammasome activation. So for example, we found it's important for the body's defense against influenza. So in this case, inflammasome is a good thing. More recently, other papers have shown that when you have too much inflammasome, it can be a overzealous response in influenza, and that could be a bad thing. Years ago, we showed that this is the NLRP3 inflammasome is important for EAE, which is a T cell mediated disease. And we also showed that inflammasomes are really critical for enhancing neuroinflammation. So the more inflammasome you have, the more neuroinflammatory responses you have. And then we also showed that saturated fatty acid and other metabolites that are associated with diseases can activate the inflammasome. And a host of labs also showed, for example, glucose and other metabolic products can enhance the inflammasome. And finally, we showed that it has a role in colitis and colon cancer. And in this case, the inflammasome affects the outcome of both colitis and colon cancer. Next slide. So to expand onto the inflammasome from cancer, because Chuan is from the NCI, so I thought I should talk a little bit about cancer. So we and others have shown the importance of inflammasome and other NLRs in a host of colon cancer and other cancers. And the top one is just NLRP3. We know that in myeloid cells prevent a colitis-associated colon cancer. All the papers I show here are from our lab, but usually there's another lab that has the same data. And we showed NLRP12, well, I'll talk about in a little bit, also affects colon cancer, and NLRX1 mitigates colon cancer. And finally, in a Nature Medicine paper, we showed AIM2, which is usually thought of as an inflammasome, has an inflammasome independent role in mitigating sporadic colon cancer. So this is one of the concepts that many inflammasomes have really important roles in controlling cancer responses. Next slide, please. And we have quite a bit of mechanism. So in this talk, one of the features is we want to find out what are commonalities of NLR proteins. So I already showed that one major function is many of them are inflammasome proteins. Sorry, could you go up, please? So another feature that's coming more recently is that many of these NLR proteins are nucleic acid binding protein, which is a little bit of a surprise. Excuse me, because when we think about nucleic acid binding proteins, we think about CGAS, for example. We think about ZBP, and we don't typically think about NLRs as being nucleic acid binding proteins. So I'll just show you some data to indicate that this may be, excuse me, an emerging theme. Next slide, please. So years ago, we found a protein called NLRX1, and this is a really interesting protein because it's found in the mitochondria. And later, Ian Wilson's lab showed that this is a RNA binding protein. And in our work published more than 14 years ago, we showed that this protein has an important role in immune response against RNA viruses. So it was really interesting that Ian Wilson was able to show that this protein can bind to RNA. And he used fluorescence polarization assay to analyze this part. Later, to the right, we showed that NLRX1 can really bind RNA as depicted by surface resonance. And this is work done by Shin Li, who's now back in I think if there's some background noise, but NLRX1 by surface plasma resonance on the right-hand side can bind to RNA, and it doesn't bind to DNA. So when we put HSV 60 DNA, NLRX1 doesn't bind it at all, but it does bind RNA. So like I say, Shin is actually back in China right now, and he's a faculty in Beijing. So next slide, please. So then I just wanted to show you another protein that we found that interacts with DNA and actually interacts also with RNA as well as RNA DNA hybrid. And this protein is called NLRC3. And on the left-hand side, you will see that we found when we express different proteins, including 2 that are known DNA binding proteins, and then take the extract and bind into biotin labeled HSV60, we can pull down the protein with DNA. So this is shown to the left-hand side, and as you can see, the DNA can pull down NRC3, IFI16, DDX41, but not NRX1 and not STING. And so then we looked at gel shift assay, which is under I, and you can see that gel shift assay and RC3 also can bind to DNA. Then to the right-hand top side, we wanted to know what is the domain that interacts with DNA. And using different domains, we were able to show in B that NRC3 LLR domain alone can bind to DNA. And we wanted to use a more biophysical assay to analyze this. So we used recombinant NRC3 in the bottom right. So these are all surface plasma resonance. And we use purified NRC3 against DNA, RNA, or DNA RNA hybrid. And the protein was able to bind to all these different forms. Next slide, please. And in addition to that, we wanted to look at if the LR domain alone can bind to DNA, and it did. So this is shown both by a pull-down assay to the left, as well as a surface plasma resonance analysis to the right. And finally, what we showed is that if we bind DNA in NLR, you have 2 different domains, or actually 3 different domains, but only 2 domains are shown here. So you have an ATPase domain, and then you have a DNA binding domain in the case of NRC3. And what we found is the DNA binding domain, when you bind DNA to that domain, allosterically, the ATPase activity went up. So the ATPase activity is shown to the left of the diagram, and you can see that when we add DNA, which is in the square line, there's a tremendous increase of ATPase activity as measured by PI release. So this shows that one domain affects the other. Next slide, please. So it was really rewarding then, a couple of years later, Hornen's lab in a really nice paper in Science showed that another NLR inflammasome, which is usually known previously not been known to bind nucleic acid, he was able to show that NRP1 can bind to nucleic acid, as shown here. And in addition, in the top left, he was able to show that it was the LR domain that was really the best at binding to nucleic acid. Interestingly, it's only the mouse NLR LR domain from NLRP1 that can bind nucleic acid, but the mouse version, which is in red, cannot. So this was just published. Then more recently from Hao Wu's lab, she used MST assay to show that NRP6 to the bottom that's labeled B and C can also bind double-strand RNA. So this, and it has a really important function in detecting coronavirus. So this is a really important paper. Then I just want to say there are other hints of NRP3, for example, binding to DNA in 2 papers in Shimida and also Zhang. And in these papers, it was shown that DNA can co-localize with NRP3. And I think further analysis would be really nice to show that this is a DNA binding protein. So with all that, I'm just trying to introduce common themes. So one common theme is many inflammasomes are NLR proteins. I think an emergent theme in my mind is several of these turn out to be nucleic acid binding proteins. And that would be really a very interesting and novel aspect. Next slide, please. So now I want to tell you about some functions that we have also detected about NLRs that's really unusual. But in almost all these papers other labs have also found the same kind of function. So some of these we were the first and some of these we were not. So intrinsic functions of innate immune receptors in T cells. Next slide, please. So I would show you 3 different proteins, but because of time, I'll probably tell you about the 2 on the 2 side. One is an NRC3, and this is a paper published a few years ago in Immunity. And what we showed is NRC3 is highly expressed in T cells. And in T cells, it can block TRAP6 ubiquination and therefore blocking FKB activation and blocking T cell activation in both viruses and autoimmunity. What's novel about it is downstream of NF-kappa B, we found that this affects glycolysis and oxidative phosphorylation. So by blocking all these pathways and blocking glycolysis and OXPHOS, this reduces CD4 T cell expansion in virus infections as well as in autoimmunity. And we showed this was inherent within the T cell population. To the very right of this diagram, that is really nice work done by Haitao Guo, who's now at AbbVie. And I've given this talk in where it's only focused on virus. And this is really nice work where he showed that the NLRX1 protein that we mentioned earlier, which was also a RNA binding protein, interacts with a protein that regulates mitochondrial function, specifically the oxidative phosphorylation function of mitochondria. So NRX1 interacts with FAST-KD5 and regulates mitochondrial oxidative phosphorylation. What was really novel is he started this whole work looking at HIV replication and found that oxidative phosphorylation downstream of NLRX1 can increase HIV replication. So when you have more NLRX1, you get more HIV replication. So this shows us 2 things. One is one potentially can target NLRX1 to reduce HIV replication. And in addition, he showed that if you can block oxidative phosphorylation with FDA-approved drugs such as metformin, you can also reduce HIV replication. And he did this not only in vitro, but also in humanized mice, where human hemopoietic system was used to replenish the depleted mouse hemopoietic system. So this has received quite a bit of attention in the HIV field, because even though the heart treatment has been very effective, there are people who start to become tolerant and the effect becomes reduced. And we suggest a simple drug such as metformin or other ways to change oxidative phosphorylation may be possible to reduce HIV replication. So in both of these cases, we're talking about NR proteins, which are innate immune receptors. And I forgot to mention the work that I'm mentioning from Haitao Guo in Nature Immunology was done in T cells, because T cells are major cell types affected by HIV. So in both of these cases, we're talking about innate immune receptors in T cells having a function in immune metabolism. So linking these 2 different types of proteins. So we think about innate immune receptor usually as myeloid cells function, but here we're showing it has important functions in T cells. And interestingly enough, it affects metabolism in both cases. And by changing T cell metabolism, they change the outcome, whether it's infection by viruses or response to viruses or autoimmunity and so forth. So the last story I will tell you then is really focused on this protein called AIM2 in the middle. And what I will tell you is AIM2 has a function in T cells. What we're showing in our paper is really the function of AIM2 in T regulatory cell. And so again, linking innate immune receptor to a T regulatory cell function has really not been done as far as I know, or at least not done much that I can recall reading much about it. So next slide. And the project started because we were looking at AIM2 as just an inflammasome. So remember I told you NLRP3 is an inflammasome, AIM2 is an inflammasome, AIM2 is a DNA binding inflammasome found by 3 different groups. And so years ago, we showed that NLRP3 is really important for EAE, which is a model of T-cell mediated autoimmunity against a myelin protein, and it's a model for multiple sclerosis. And when the mice do not have NLRP3, you get very little EAE, suggesting that this is an exacerbating factor for multiple sclerosis-like symptoms. Other people shown to the right and left, Inoue and Lalore, have shown the same. That is, with ASC knockout and NLRP3 knockout, there's very little EAE or caspase-1 knockout. So we just expected, if we look at AIM2, that if we have AIM2 knockout, that we would have exactly the same data. In contrast, what we found is the opposite. And this is shown on the bottom left. So we took AIM2 knockout, which is the red line, versus wild type, versus caspase-1 knockout, caspase-11 knockout, which is called ICE, or ASC knockout. They're both at the bottom. And in these experiments, the ASC knockout and ICE knockout have absolutely no clinical score, very little clinical score, if any. But if you look at the AIM2 knockout, which is the red line, you have a really high clinical score that's even higher than wild type. So this suggests to us that in this case, paradoxically, AIM2 has the opposite function of ASC, which is not what we expect, because AIM2 usually requires ASC for its function. So we suggest that AIM2 has an inflammasome independent function. Indeed, we look at demyelination and also inflammation in the EAE mice, and we found that demyelination was greatly reduced in ASC knockout as expected, and inflammatory foci was also reduced. But both of these are greatly enhanced in AIM2 knockout. But if we look at inflammasome biomarkers, IL-1 and IL-18, we see no difference between AIM2 knockout and wild type. Next slide, please. So this clued us in that there may be a different function that we're looking at. So we look at CD4 T cells in this mouse model because this is a T cell model. And what we found is IL-17 and interferon positive cells are increased in the AIM2 knockout. But if you look at the right-hand side, the T regulatory cells are reduced. So there's a skewing in AIM2 knockout towards effector cells and against immunoregulatory cells. Next slide, please. And so at this point, we started wondering, since we don't think people really have focused on AIM2 in T cells, we look at its profile in T cells for MGen or EMBL. And if you look at T regulatory cells, you can see in the mouse database, there's quite a bit of AIM2 expression, pretty respectable. And if you look at the right-hand side, the human AIM2 expression is really pretty high in regulatory T cells. And this has to be taken account when you can go down and look at inflammatory macrophages. They express probably about the 10th level of AIM2 as regulatory T cells. So this came to us as a really surprise because we weren't really focused on T cells when we were studying AIM2. Next slide, please. So then we wanted to know if the T cell AIM2 function is intrinsic within T cells. And we did this two ways. One is we took wild type CD4 T cells and transferred them into RAG1 knockout. And in comparison, we took AIM2 knockout CD4 T cells and transferred these into RAG1 knockout. We subjected these RAG1 knockouts with transferred T cells then to EAE and measured the clinical score. And as you can see, the AIM2 knockout T cells alone can induce more clinical score compared to the wild type. If we look at this population, what we found was the AIM2 knockout, the recipients of AIM2 knockout T cells have lower amounts of CD4 FOXP3 T regulatory cells, therefore linking the function of AIM2 possibly to Tregs. Next slide. In addition to this model, we also look at a different model because we didn't want this to be a single model issue. So we look at T cell mediated colitis. And in this model, you can introduce wild-type naive T cells, or you can induce wild-type naive T cells and T regulatory cells. And the T regulatory cells will mitigate the disease. Or we can add in AIM2 knockout T regulatory cells, which we presume are not working very well. And we would expect it not to reduce, not to alter the disease. So indeed, that's what we saw on the right-hand side. So the the green line is naive T cells alone, and you see a loss of weight because these mice have colitis. Then if we add T regulatory cells from the wild type in the blue line, we're able to mitigate this disease. However, we add AIM2 knockout T regulatory cell, it's not very functional. So it wasn't able to really mitigate the disease. So to look at the role of Tregs more specifically, we knock out AIM2 only in FOXP3 cells. Next slide. So I should say that this is done with Zhengli Guo and Yisong Wang at the University of North Carolina. And they have helped us a lot. And Emily Chow is the first author on this paper. So what we did is we did an AIM2 specific knockout with the help of Yisong's lab. And this is shown here on the left-hand side. And to the right-hand side, what we show is if AIM2 is specifically reduced in FOXP3 positive cells, then what we have is increased EAE. So AIM2 really has a function in FOXP3 positive T regulatory cells. And then in the lower part, if we look at this mouse and look at the amount of T regulatory cells, we see that FOXP3 positive cells are decreased to the left, and we see that interferon positive cells are increased. So exactly what I was telling you before, that is in AIM2 knockout, if we knock it out in Tregs, we see a drop in Tregs and we see an increase in interferon gamma positive cells. Next slide. And we also did a cell fate mapping analysis. And although there's some question about how tight this assay will be, we actually used Jeff Bluestone's mouse in this study. And so you can follow the fate of these mice. And basically what you have is you have FOXP3 driving GFP. But if this is a transient expression, the cells will not express GFP, but you can go have the Cree induced from the same promoter, FOXP3 promoter, and Cree will take out the flox on the second R26T gene to take out the stop codon and allow the expression of TD tomato. So as long as the cells have expressed FOXP3 to induce Cree expression, it'll express tomato, TD tomato. But if it's the FOXP3 is transient, then GFP will not be expressed. So this is a way to see if a cell has previously been positive for GFP, remain positive for FOXP3 expression. So what we looked for is this the red cells, and this is on the bottom left. So we're looking at the red cells, and what we found is these TD tomato cells actually do not express FOXP3, and there's an increased amount of these in the absence of AIM2. So what this suggests is that AIM2 is there to remain and maintain the stability of T regulatory cells. Next slide, please. So then we wanted to know what is the function, how this AIM2 functions in T regulatory cells. So we did a RNA-seq of T regulatory cells from AIM2 knockout versus wild type, and using a gene enrichment assay, gene set enrichment assay, we were able to find that in the absence of AIM2, we have much more heightened mTOR, PI3K, and AKT pathway. It also has heightened glycolysis and increase of c-MYC. And these can all be done, verified by Western blots shown to the right-hand side. So we see increased c-MYC in the presence, in the absence of AIM2, increased mTOR downstream signals such as S6 or 4-EBP, and we also see PAKT phosphorylation. Next slide, please. And so we also did seahorse analysis, looking at the immune metabolic changes. And what we found is if you look at AIM2 Tregs induced in vitro, what we found is that in the absence of AIM2, we have increased glycolysis, reduced oxphos, and really significantly reduced fatty acid oxidation. And all of these phenotypes are consistent that we have an alteration of T regulatory metabolism. So in the absence of AIM2, we have an immune metabolism that's opposing of T regulatory cell development. And in the presence of AIM2, we have a phenotype that's more consistent with T regulatory cell immunometabolism. And this metabolism has been nicely shown, for example, by Hombo Chi and also by Erica Pierce, etc. Next slide, please. So then we wanted to know what is the mechanism of this. And we did a mass spec co-IP with Shan Chen at our university. So we took AIM2 knockout CD4 T cells and wild-type CD4 T cells and treated these with TGF-beta and then brought them down with AIM2 antibody and looked for differences. And what we found to the right-hand side top is that AIM2 was able to bring down RAC1. And so what does RAC1 do? RAC1 actually recruits a phosphatase, PP2A, and this phosphatase and then can dephosphorylate AKT. So in our mind, AIM2 would recruit RAC1 and PP2A, dephosphorylate AKT, therefore inhibit the mTOR pathway. And that's what we're looking for down at the bottom left. We showed that AIM2 indeed can interact with RAC1, and we did this endogenously, and we were able to IP AIM2 and look for RAC1 interaction. We did this in two directions. And then we wanted to know how does AIM2 affect PP2A and RAC1 association. So in the wild type, we were able to see that RAC1 and PP2A do associate. This is the bottom right panel. We were able to see that the two do interact, but in the AIM2 knockout then, what we found is RAC1 and PP2A interaction was greatly reduced in the absence of AIM2. So what this suggests is that the absence of AIM2 actually causes RAC1 and PP2A to not associate, therefore allowing AKT to proceed and phosphorylate. Next slide, please. And so to really determine if this is the case, we did an overexpression experiment in T regulatory cells. We added in PP2A and RAC1. So the wild type plus vector is just wild type Tregs with empty vector, and wild type plus overexpressed RAC1 and PP2A are introduced by lentivirus. And as you can see at the bottom, we also assayed for PAKT phosphorylation. And this is a log phase. And we were able to show that there's a shift in PAKT phosphorylation if we overexpress these two regulators that can down-regulate PAKT phosphorylation in wild-type cells. However, in the AIM2 knockout cells, in light blue and brown, when we overexpress these two proteins, it has no effect, suggesting that AIM2 is critical for the regulation of AKT by PP2A and RAC1. And in vivo, if we go to the right-hand side, looking at EAE spinal cord, we show that if you don't have AIM2 and T regulatory cells, the left is just a flow diagram, and on the right-hand side is a geo mean calculation. And you can see that in the absence of AIM2, there's an increase of PAKT. Again, AIM2 is a negative regulator of PAKT. Next slide, please. And finally, what we were looking at is what is inducing AIM2 in T regulatory cells. And I just want to make a really fast conclusion that it turns out at least TGF-beta can negatively regulate AIM2. Recently, there's another paper showing that IL-10 can also not negatively regulate AIM2, but TGF-beta induces AIM2 in T regulatory cells. And IL-10 can also do the same. So in T regulatory cells, all these cytokines that can upregulate Tregs can induce AIM2. Next slide, please. So in conclusion, what we found is a different function for AIM2 in T regulatory cells that can actually affect immune metabolism. So to the right is when you don't have M2. And when you don't have M2, RAG1 and PP2A cannot come together. They cannot block AKT phosphorylation. So mTOR proceeds, and mTOR proceeds to induce c-MYC. And then what we have in that case is increased glycolysis, decreased oxphos, decreased fatty acid phosphorylation, and then at the same time, decreased T regulatory stability. So what we have shown then is 3 different cases where we're talking about innate immune receptors affecting T cell function in a pretty profound way by changing immune metabolism. So now I want to go to another function of many of these inflammasomes or NLR proteins. Next slide, please. So I already concluded for this. Thanks. Just next slide, please. So there's a lot of work done on innate immune receptors and microbiota. Can I have the next slide? So, and we have published a review in JEM summarizing many of the data. But what I want to talk about today is briefly about NLRs that are affecting microbiome. And the one that we studied the most is called NLRP12. So this has been found to have inflammasome function in some circumstance, but in many cases, it seems like the absence of NLRP12 results in increased signal transduction and activation. And this has been shown by several groups. And several NLR proteins actually have this type of function by inhibiting NF-kappa B or by inhibiting interferon pathways. And so the NLRP12 in our hands decreases non-canonical NF-kappa B, which is shown in the next slide. So this slide simply shows dendritic cells that we use. The top left is dendritic cells that we activate from wild type or NLRP12. And we're looking for non-canonical NF-kappa B activation, which is viewed as P100 processing to P52. So the NLRP12 knockouts, you see increased P100 to P52. So in our mind, NLRP12 inhibits this process, and the absence resulted in increased activation of NF-kappa B. So if this is a negative regulator, we would assume that if we take out the gene, we would have more colitis, and that is indeed what we found. So the middle panel with DSS in the colitis model, we found the NLRP12 knockout actually have very low survival. They have tremendous inflammation. And then when we look at a colon cancer model, they have lots of polyps as shown by the colon with the arrow. So this shows that this is a negative regulator of inflammatory response and a negative regulator of colitis. And if you don't have this gene, you have really flagrant colitis. Next slide, please. So years later, we looked at this again, and this is work by Leo Chen, who's now also at AbbVie. And what he found is an specific pathogen-free facility, and NLRP12 didn't do as well, but it wasn't as sick as what I showed you before. And what intervening time, our mouse facility was changed to a much cleaner facility. So we wondered if the microbiome was involved. And we re-derived these mice in germ-free facility. Now we don't see any difference between the two. So next slide. Through a number of studies, we were able to show to the bottom that NLRP12 knockouts in the red have reduced expression of several different orders of bacteria and a specific family of bacteria. So in the bottom part, if you look at the left-hand side, we used litter mates, and we also got mice from different vivaria to make sure it's not vivaria specific. And litter mates, of course, is the best kind of study. And what we showed is that the NLRP12 knockout have reduced expression of many of these bacteria. And it turns out Lactosporiaceae was what we focused on. Next slide, please. So what we wanted to know is if Lactosporiaceae has any beneficial effect. So if this is missing in an NLRP12 knockout, mice that have really strong colitis, does this bacteria have beneficial effects? So we fed the bacteria back into NLRP12 knockout mice. Excuse me. And what we found is that the NLRP12 knockout mice fed with bacteria did much better and didn't lose as much weight. So this bacteria can be almost medicinal. I'm sorry, I have a really bad allergy cough that always comes about when the air condition comes on or the heater comes on. Next slide, please. So we were very interested in this bacteria and we did a lot of work with it. But what we didn't expect was its importance in radiation. And so to segue into this, we know radiation can be good where you have cancer therapy, or it can be bad where you have accidental exposure, such as in Japan a few years ago, or a nuclear bomb, for example. So in those cases, what we're mostly worried about is down below hematopoietic syndrome and GI syndrome. So next slide, please. So I just want to introduce one thing that is back to the inflammasome. We have a paper that is either coming out or coming out soon. And years ago, in terms of the inflammasome, it was found that AIM2 inflammasome actually exacerbates inflammation. This is a paper published in Science in 2016. But more recently, we and another lab have jointly looked at inflammasome and radiation, and we actually couldn't find any detrimental effects of the inflammasome. And to the A is we looked at exactly the same mice, AIM2 knockouts versus wild type. And there's, in fact, if anything, the presence of AIM2 was protective. And if we look at to the right, caspase-1 knockout or ASC knockout, again, the presence of the inflammasome was protective. But interestingly enough, it was protective mostly in the males. And the reason we think it is different is because both of the latter papers use litter-made controls, and that could be the reason for this difference. But this also marked that perhaps different facilities have very different microbiome, and therefore these microbiome can affect the outcome of inflammasomes. So we've been just thinking about this. And next slide, please. So we were wondering, how does inflammasome play a role in radiation sickness? And so Hao Guo in the lab was looking at radiation, and she noticed in the top left that when we radiate mice with near lethal radiation, a small percent of the mice can survive. In this case, 3 out of 20. And she wondered if these mice have a different microbiome composition, and she profiled the microbiome, and indeed they were different, as shown in B. Next slide, please. So she did a really very nice study by taking fecal materials from these survivors, which we now call elite survivors, and also took them from control mice of the same age. So these are pretty old mice, and took the fecal material. She transferred them into germ-free mice as a recipient, and then exposed these mice to radiation. And what she found is the fecal material from the lead survivor in red was able to protect germ-free mice recipients from radiation-induced death. And this is shown in I, and the clinical score is shown in J. So she was able to find that there's microbiome indeed that can protect mice from radiation sickness. Next slide, please. And so what she looked at is the microbiome difference between these elite survivors and age-matched controls, and found that the microbiome were very, very different in K. And then in M, she was able to show that mice that received fecal material from the elite survivors have increased numbers of these bacteria shown in the red. And among these bacteria was Lactobacillus, our old friend, suggesting maybe Lactobacillus, as well as these other bacteria, might be able to protect against radiation damage. Next slide, please. So what she did is she transferred these bacteria into mice and basically engrafted these bacteria for a total of 8 weeks and then subjected the recipient to radiation. And what she found was, if you look at B, mice that received lactose-free ECA did best. So even though some of them still died, but about 60% of these mice actually survived compared to control, where only about 10% of the mice survived. So that's a really big difference. In addition to lactose-free ECA, she found that Enterococcus had the same effect, and lactobacillus, which is found in yogurt, had the same effect. And we found many effects of these bacteria, including effects on the bone marrow replenishment and also in the GI. So this is shown to the right. So the GI is healed much better in lactose-free recipients. And this protection amazingly lasted for over 300 days. Next slide. So if you can just move to the next one. Thanks. So we wanted to know, when does this happen? When do the beneficial bacteria get expanded? So what she did is we didn't know the outcome of the mice, but we took a lot of mice and radiated them and looked for expression or the amounts of lactose-free ECA or Enterococcacia. And what she found is that day 1 of radiation, in the elite survivors and non-survivors, there's really no difference in lactose-free ECA. But if you look at day 7, the ones that eventually survive have a significant increase of lactose-free ECA, which is further increased by day 21 and day 30. So this suggests that for whatever reason in the elite survivors, there's expansion of the beneficial bacteria after radiation. Next slide, please. And if we look at patients who received whole body radiation, received radiation, these are patients from Duke. What we found is that the patients with diarrhea that lasted less than 10 days, indicating they have less damage, have increased numbers of lactose-free ECA, Enterococcacia, lactobacillus. So again, these 3 bacteria were associated with better outcome in terms of diarrhea. Next slide. And in addition, we wanted to know what is downstream of this bacteria. So lactose-free ECA is a well-known bacteria that makes short-chain fatty acids. So we were able to separate lactose-free ECA that make low amounts of short-chain fatty acid in blue or high amounts of short-chain fatty acid in red. And we put these 2 groups into mice and then radiated these. And what we found is the bacteria that have high short-chain fatty acid was able to protect much better than those that have the blue one. Next slide, please. And then in addition, what we found is that we just put the short-chain fatty acids in, we were able to protect these mice from radiation-induced death. So especially if we put propionate, which is the solid red line, we were able to protect these mice from radiation-induced death. And a group in Michigan actually has previously shown that butyrate was effective in protecting against radiation-induced damage. Next slide. In addition to, could you skip to the next slide? So looking at the mechanism then, what we found is in the mice that received the propionate, indicated here as P or B butyrate or control, what we found is in these mice, we were able to show that tissues derived from these mice, from mice treated with propionates, have decreased amounts of H2AX, which is an indicator of DNA damage. And in addition, if we look to the right, I think one is bone marrow, that's right. One is GI, one is bone marrow. P2AX was reduced in the presence of propionate and butyrate, suggesting some mechanistic changes. And in addition, we also saw reduction of ROS, reactive oxygen species. And furthermore, propionate on the bottom right was able to protect these mice for over 300 days. So this is pretty remarkable early feeding of propionate to the mice protected them for over a year. Finally, next slide. We wanted to know if there are other metabolites that could also be protective. So Kun Lu at our university would, and his student was greatly helpful in doing non-biased metabolome analysis. And in the metabolome analysis, they were able to find in the middle that tryptophan was greatly increased in the elite survivors. So what Hao did was treat these mice with two tryptophan metabolites in red, and she was able to show that these tryptophan metabolites was able to protect mice against total body radiation damage. So next slide, please. So finally, what we summarize from this is we know that innate immune receptors, excuse me, such as NLRs, can alter the outcome of gut microbiome. And we showed that one specific microbiome, one specific bacteria, Lactobacillus, was effective in protecting against radiation sickness as well as colitis. And this is through short-chain fatty acid. But in addition, we found that tryptophan metabolites can also protect against radiation damage. Last slide, please. So in this talk, I basically shown that innate immune receptors have so many different roles, including, of course, infection, inflammation, and also in T-cell mediated autoimmunity, metabolic diseases, cancer, vaccine response, which I didn't talk about, microbiome control, and more recently, T-cell immune metabolism. So this has a lot of important functions that we have yet to really fully appreciate. Finally, the last slide. So I just want to acknowledge the people, and I mentioned to the right, Wei Chang Chao and also Hao Guo, who did the two studies I mentioned. And then June did the study on radiation with the inflammasomes. And to the left-hand side, Xin and Meng, I mentioned them, did the NLRC3 work. Haitao Guo did the NLRX1 work. Elena Rampinelli really introduced us to the immune metabolism analysis. And Toru and Yoshi did the NRC3 work, and we were greatly helped by Yisong and Zheng Li, Shan Chen, and Li Shan. And at UNC, we're helped by Belfer Sarder, Jason Whitmire, Kun Lu, and his wonderful graduate student, Yoonja Lai, who's now at Columbia, and others listed and from the University of Michigan, Duke, and Cornell, our colleagues. And I just want to thank you very much for your attention.
Thank you so much, Jenny. That was cover a lot of ground. I think there's going to be a lot of questions. So please raise your hand or just put your question in the chat box so I can read out your question to Jenny. So I guess first question is from Yangxin. Yangxin, you want to ask a question yourself?
Oh yeah, just Jenny, what is any difference between C-gas and M2 for DNA sensing? Is any structure different?
Yeah, that's a great question. Something we're actually really looking at. I don't know if Xin Li is on the, but something we're really interested in. Like when you have weird structures, do they differentiate between the two? We don't have the answer yet. So far, they look like they're recognizing all the same things. Maybe one recognizes one form a little better than the other, but really it's hard to quantitate. But that's a really good question. I should say that, I should really mention your paper. So in your paper, the microbiome in the colon cancer had very different outcome than our microbiome in non-colon cancer. So I think there is a little bit of concern when we're using these same microbiome in colon cancer because that's a really complicated situation. But, I just want to mention that. But because of time, I didn't go through our cancer work.
Okay, I think next question is from Sean. Sean, you want to ask question yourself?
Yeah,
Go ahead.
a very interesting talk. I'm wondering whether is DNA required for the AIM to function in Treg cells, or just
That's another great question. No, we are looking at that right now. So we have a project in the lab that's looking at whether the DNA sensing component of AIM2 is really required to Treg function, or is entirely different. So we have some data, but not in terms of linking the two functions yet. And there are a couple, there's not very many papers on DNA sensing and T cell response. And there was a recent paper in immunity. And I don't know if the authors might, it's really hard to look at all the audience, but a really nice paper showing that DNA sensing in T cells, and it's a paper from China, a T cell is important for T cell proliferation, for example. So we're definitely borrowing from that paper and looking at this question.
Thank you.
Well, I haven't seen Bing for a long time. So Bing, go ahead.
Yeah, Chen, thank you. Hi, Jenny, this is Bing. So I'm from Shanghai.
Yeah.
Very nice talk. Can you hear me? I see you freeze my screen. I don't know whether Jenny can hear me.
I can see you, yeah.
Oh, you can. Okay, thank you. So my question is, I know you utilize the irradiation to induce the gut damage. So wondering, when you compare the phenotype with normally people using the DSS treatment, the phenotype in your like knob mice and other model has any major difference? And also, would that indicating there's a differences in the mechanism for induced inflammation in the gut?
Yeah, so in the NLRP12 knockouts, we did use lactose-free ECA, and it was pretty beneficial. And what we found, it was if we use short-chain fatty acid, we can have the same outcome. So in those cases, both the bacteria and the metabolites seem to be the same in mitigating these effects. So I think in those cases, they may be pretty similar. How is actually looking at some of these issues right now in terms of, how exactly, which receptors are involved, for example, what mechanism is involved, and so forth. So we are, and she's going to dig much deeper into the mechanism. But at least superficially, but bacteria is the same.
It's the same, right?
Same. We haven't looked at HDAC, for example. Is that one possibility? And that's known to be downstream of butyrate. So those are some of the mechanisms that we're exploring.
Right. Thanks. Nice to see you, Jenny.
Same here. Lots of great questions. And the good thing is we're answering all those questions in the lab. So we must be hitting on the right questions. Yeah, Peng Yang.
Yeah, Peng Yang.
Yeah. Peng Yang, go ahead.
Yeah, this is Peng Yang.
Yes.
Can you see? Hi, Jenny. It's very nice to see you. I was postdoc in Duke University.
Oh, cool.
10 years ago, and
That's
now
great.
I'm teaching. Yeah, very nice to see you. I have attended your seminar. Very nice. Always very nice talks. Yeah, so very good. Yeah, so it's an impressive talk. I have a question on the second M2 on the Treg cells. So is there any some difference on the inflammasome in the Treg cells or effector cells? Yeah, so I think it's very interesting. Yeah.
Yeah, we are looking at that because really in T cells, you don't see much IL-1. T cells don't have much IL-1.
IL-1?
So it wouldn't be that I know of. If you look at message, IL-1 is hardly there.
Okay.
So I don't think it's the typical inflammasome pathway. It could be, one thought I had is maybe affecting caspase-1 and maybe affect other downstream signals. So that's entirely possible. but we know
Oh, okay.
the typical IL-1 that we think about.
Okay.
Usually you think about inflammasome, it's always IL-1 or IL-18, right? We don't see
Oh,
any of that. So we don't think this is the typical inflammasome, if it is an inflammasome.
oh, okay. Yeah, very nice. So yeah, another question is that sometimes that FOXP3 Treg cells will transfer to the RA gamma T positive T17 cells. Yeah, have you seen any yeah, evidence on that?
We saw some. So in some cases, we see Tregs reduction. So the cell-fate analysis, it didn't show IL-17. Instead, it showed interferon gamma.
Okay. Oh,
So the
okay.
T-fate analysis, we show. So I've given this talk at a few places, and some really, the FOXP3 experts would say, well, the cell-fate mapping may not be exactly tight, except that's Sasha Rudensky's mice, right? Has that problem. We use Jeff Bluestone, so I don't know if his mice have. It's not reported to have the same problem. But we're not FOXP3 people. We're not Treg people. But you saw, it's a really great T cell biologists who helped us with these studies. So those would suggest that what we have is reduced FOXP3, but increased interferon gamma.
Oh, interferon gamma. Oh,
yeah.
Yeah, that is very wonderful work. Thank you.
Oh, thank you. So it may be a skewing, maybe just changing the metabolism and switching the cell type.
Oh, okay. Yeah.
I think the next question from Bing Chi, you go ahead to ask question.
Okay. Jenny, thank you very much. I have learned a lot, really.
Oh, you're very.
Yeah, I observed that you have mentioned that there are a big gap between the male and the female mice connect with relation. So whether you have observed difference of their microbiota between this thing.
We must have some connection because those are exactly the studies that we are doing. I almost feel bad saying that over and over. That is exactly what we're thinking. Why would, and it's really weird in that it's affected by the sex of the mice, right? So, that was hormonal, one would think. We actually didn't believe the data. So poor June, I forgot to mention her name, June Brickey did all of those studies. And some of the samples, we have like 50 mice. So we just didn't believe this data. And she repeated the experiment so many times. So now we are profiling microbiome, seeing if we can see protective microbiome that we can then transfer again.
Okay, we appreciate your next paper about it.
Oh, thank you.
Yeah,
I think it'll be a really cool paper. I did communicate with the WHO paper because that was from Richard Flamel's lab. And the only conclusion we can come up with is maybe his facility has some really unique microbiome that made P3, to be a damaging factor, because neither at Duke nor at UNC did we see this.
Okay, thank you very much. Yeah.
Unless, as many of you know, Duke is 12 miles away. So unless there's something unique to the environment in North Carolina, that we're generating some special microbiome or something.
Okay, so next question is from Zheng Yang.
Okay, I'm Zheng Yang. Thanks for your excellent speech. I have a question. Does AIM2 have a similar function in myeloid cells as in Treg?
Yeah, that's the close to the question that was asked. Not exactly the same question, but in myeloid cells, AIM2 is inflammasome. There's no question. That's the main function.
Yeah.
In Treg, when we looked, we don't see cytokine. We don't see like typical inflammatory cytokines, which is expected because you don't expect inflammatory cytokines such as IL-1 or IL made by T cells, right? Instead, the main function seemed to be the mTOR pathway. So we
Okay.
think they may be very different. We also saw some interferon changes, by the way. So that was pretty interesting. We have a project going on looking at that. But AIM2 and T cells and myeloid cell, as far as superficially we can tell, they look very different. We're looking at that right now to see if there are other things that may be shared. But this is actually not surprising because we had a Nature Medicine paper, and the Kanaganti lab also had a cell paper on AIM2 and cancer. And that is also on mTOR, on MYC, and on, was it DNA PK? Yeah, DNA PK.
Okay.
So in cancer, so just to put it into perspective, AIM2 was found initially in cancer, right? Before we ever thought it was an inflammasome protein. In cancer, it has really a lot of functions because that's why it's called absence of melanoma. AIM stands for absence of melanoma. So AIM2 is actually not found in some cancer cells. So people think it's a tumor suppressor, and that would agree with what we're finding, right? Without AIM2, you have increased MYC. So if it's absence in cancer, you would have increased MYC, you would have increased PI3 kinase, you would have increased proliferation. So this could be entirely separate from the inflammasome, but we don't know for sure.
Okay, thank you very much.
Sure.
Well, you see, Jenny, usually our seminar, the speaker get bombarded with the questions. Usually, sometimes the question section could be longer than your real seminar. I think, okay, so I think I'm going to ask a few questions. First thing is, I'm actually curious about, you know, the expression of AIM2 in the inducible Treg cells and n-Treg cells. Have you ever heard that?
So I didn't have time to mention those, but we actually look at both of those.
Right.
And the whole paper, almost everything that was done with both inducible Treg and just like we did it both with in vivo Tregs isolated and in vitro induced Treg. And everything was identical. So at first we thought maybe it's functional in one population, not the other, and there's no difference. So we think it's the function of AIM2 is not differentiated between those populations. You know, it's just found in Tregs overall.
But when you actually describe the machinery of AIM2, you describe that it's actually interact with the mTOR pathway, but it's also because it's promote the phosphorylation of AKT through the PP2A. And you know, you actually in your illustration, you also show the downstream with FOXO protein. So I'm actually curious because FOXO protein obviously is an important part of the mTOR signaling package. At the same time, FOXO1 can directly participate FOXP3 expression. So which pathway you think is more dominant in this aim to regulate Treg cells signaling?
You mean mTOR versus FOXO?
Right.
Yeah, I mean, that's a really good question. We didn't really focus on FOXO, but we really do see a difference in FOXP3 expression.
Okay.
You know, so it's possible it's on both or converging. We did not differentiate. As you know, working with Tregs and working out molecular mechanism was not the easiest thing. We did everything with real cells. You know, nothing was done in cell lines. So it was, so we didn't really, we just, at the end, we just kind of didn't deal with that other part of the pathway.
I see.
But the reviewers didn't push us for it. So we were really happy because it took us forever, took us a year to revise the paper just on the mTOR part.
Right.
So that's a really good catch. When we went to the mechanism of exactly what happens, most of those were with inducible Treg, you know, the biochemical, of course, the biochemical mechanism. We came back and tried to verify some of those in an EAE model to show that if we use EAE model and looking at FOXP3 specific AIM2 knockouts, we were able to see changes in AKT.
I see.
But most of the regulation, the PP2A and all that, those were all inducible.
Right, right.
Yeah. The phenotype was done in both.
I see. Okay, okay. I think my third question is related to the third part of talk about NRP12. I
Yeah.
guess I still remember in your Nature Immunology paper, you're describing the microbiome part. And in the end, you did the bone marrow transplantation, and then all the phenotype with the bacteria is due to, I think, TNF, if I remember that correctly. The DSS colitis part. IL-6, sorry, IL-6 and TNF.
IL-6, TNF, and also IL-1 actually.
Yeah, exactly.
So but we only look at those three. And then when we blocked TNF and IL-6, we were able to alter the microbiome. So the paper, basically, the conclusion was, and it wasn't very popular at the time, but now I hear many more people saying that this is probably what happens. So we have a genetic effect, right? The NLRP12 gene. And then we have a microbiome effect, which is environmental. We typically think of microbiome now as environmental. And the two interact and they go in a circle, which was very confusing for the reviewer. And so what we found is that the NLRP12, if you don't have that gene, you have increased inflammation. As a result, you have increased IL-6 and TNF. If you can block those two, you can you can replenish the good microbiome.
Right.
So inflammation is affecting the microbiome. Then we found if we replenish the microbiome, we can also reduce inflammation. So it's a two-way system.
Sure.
That is not so straightforward, that it only goes one way. So that's the part that we had such a hard time convincing the reviewer. And now a lot of people would say, sure, that happens, but back then it seemed like this was, can't remember, 4 years ago, 4 or 5 years ago. So the concept was, you have a linear concept.
Yeah,
but
sure.
It's really a circular concept. And we're still trying to figure that out. We're still very puzzled by some of the genetic link to microbiome and vice versa, to be honest.
I guess my point is because in your radiation story that you're describing a similar microbiome, but it's actually caused by different machinery due to the short-chain fatty metabolism. So what do you think about the short-chain fatty metabolism in this DSS scenario?
So in the DSS scenario, we also we actually did the short-chain fatty acid in an obesity model.
Right.
And there we were able to show these short-chain fatty acids also reduce IL-6 and TNF.
So you think that the final consequence or final readout is going to be short-chain fatty acid, or it should be the pro-inflammatory cytokines in terms of DSS scenario?
In the DSS scenario, probably the inflammatory cytokines are the final, really damaging part.
But you
because
still believe that the short-chain fatty acid is somehow medium product, which actually caused the consequence of pro-inflammatory cytokine?
That's what we showed in vitro.
I see.
We showed in vitro, if you just treat cells with short-chain fatty acid, TNF, IL-6 are reduced.
But that's more target to the myeloid cells and macrophages.
Myeloid cells, exactly.
I see. Okay. But in your irradiation story, you're talking about more, I guess it's more cell damage and stem cell part?
We're still looking at that. In fact, we just got a bunch of stem cell markers that we're looking at, intestinal stem cells. So the question is, we see an effect both with IE, which I didn't have time to get into detail. It has both effects on hematopoietic bone marrow cell progenitor cells. So we show that progenitor cells greatly is increased. In addition to that, we also showed IEL cells, intestinal epithelial cells are also altered.
I see.
But we don't know what are the signals, like what exactly are the signals of either tryptophan or short-chain fatty acids. In short-chain fatty acid, there's some mechanism that could be explored, for example. So gene expression would be one. So we haven't gone back and looked at if, for example, propionate would change HDAC on chips an IL-6 promoter and TNF promoters. That would be the ultimate of
Right.
looking to see. And all of this has to be done in primary cells.
Oh yeah, sure.
Yes.
I guess I have just a little last little questions, because I noticed that when you treat the mice during DSS with the beneficial microbiome, you do rescue your NLRP12 knockout mice, but in the wild type mice, I don't see that much effect.
Yes.
Can you comment on that?
Yeah. So our explanation, which may or may not be correct, is that the wild type already have lots of lactose-free CA. We profiled them, right? They have plenty of lactose-free CA. So it doesn't have a hole of lactose-free CA. So that's why even when we add it in, it doesn't have any effect. But the NLRP12 knockouts has a hole in lactose-free CA, so it fills that niche. So it goes in and can fill the niche. Now, in the same study, in the short-chain fatty acid and obesity, what we found is if we use short-chain fatty acid instead of bacteria, that has a beneficial effect in both wild type and P12 knockout. So here, you're not replacing bacteria. You're not getting bacteria in to compete anymore. You're just using a metabolite. And what was pretty remarkable was, I think when we use either the bacteria or short-chain fatty acid, we were able to reduce obesity, like the adipose fat content, from like 20 something to like down to 10. So it was a significant reduction of fat content in the body. So that's our explanation, that wild type, there's no more niche. The lactose-free niche is already filled. It already has a competitive advantage. And so you put it in, it's not going to expand anymore.
You think there is a saturation level of certain metabolites, which actually...
Short-chain fatty acid, we didn't, we added in, it did have an effect regardless of whether it's
I see.
wild type or P12 knockout.
Interesting. I see. Okay.
So that's explanation.
I see. Okay, thanks. I think the next question is from Xiaoqing. Do you want to go ahead and ask question?
Yes. Thank you, Jenny. Very nice work. I just have 3 naive questions. So the first is, you said there are some NLR protein, they recognize RNA virus and some recognize DNA. So,
yeah.
And both using the LRR domain. So I want to ask, there are any difference in those RNA receptor and the DNA receptor, and are they localized in different cellular?
Yeah, those are great questions. You guys must be like working in my lab or something. So we think that an issue that really hasn't been studied a lot in our field or in a lot of innate immune receptors is the concept of where these proteins are. For proteins such as MAVs or CGAS, Ting, there's a lot more studies. For NRP3, there's quite a bit of studies too. For example, different localization, Golgi, ER, and so forth. For the nucleic acid binding ones, we really don't know where they are binding nucleic acids. The nucleic acid binding concept is actually pretty new. Even though I mentioned NRX1, for some reason, it never received much attention. And that's the one that's in the mitochondria that we discovered that Ian showed it was an RNA binding protein, and we showed it was an RNA sensing pathway. But when we published in RC3, it was still the second one, and it wasn't, I think people were still wondering, what does it mean? I'm not sure what the view is, but the 2 recent paper with NRP1 and NRP6, and I should say for all 4 of these, not only are lots of DNA binding assays used, but more biophysical assays were used in every case. So I think there's quite a bit of credibility And we're exploring this with other methods to look at their nucleic acid binding. But the question is, where do they do this? You know, do they do this in a specific compartment, or do they recognize nucleic acid only when they're activated or non-activated, or what is the, where do they do this, or what happens after they recognize nucleic acid? I think that's completely open as of now. You know, the recent paper from Hao Wu showing NLRP6 binding to RNA is really nice, and she showed the importance of that and partition of the protein. And so that was a really nice paper. And I think we can learn a lot by looking at, after binding to DNA or RNA, do they change where they're partitioning? Do they go into different compartments that are enriched in lipids and so forth? So those are really interesting questions.
Thank you. And my second question is, do you know there is any reports about there are any NLR mutation in the human clinical autoimmune disease? Because
yes,
yeah.
Yes, that's what started this whole thing, really the whole field. So C2TA, the reason it was found was because if you don't have C2TA, you don't have class 2 MHC. And there are patients without C2TA and they die when they're really young because they become really immunodeficient. So that was the first link of NLR proteins. When this before C2TA was even thought of as it was before the NLR family was discovered. And later for NLRP3, the reason that was found to be so important in human is because there's a whole class of proteins. There's a whole class of disease called familial cold associated autoinflammatory syndrome, FCATS. I think that's what it stands for. And Hal Hoffman was the first one to show that this family of disease is due to mutations in NLRP3. And that's how the whole field came about. Then Dan Castor and Mike McDermott, I think, all found the same thing in patients. So that's how this whole family became so important. We know they have a function in humans. And later, several of these proteins were found when they're mutated can cause disease. So that's a NOT2 is a really good example. So that's linked to Crohn's disease in Caucasians, but probably not Asians. So Crohn's disease linkage to NOT2. So this family is highly associated genetically with diseases.
Okay, thank you. And the last question is about your last part. So do you consider using this specific microbiome using to the cancer radiotherapy, the clinical?
Yeah, for to look into during cancer therapy, if patients have damage, if we can use this to mitigate it.
Yeah.
Yeah, so we have a young resident who's now become faculty, who's putting in an IND to see if we can, test this. Like I say, there may be caveats because Xinfu has a paper, I believe, in immunity about being cautious, especially when it's in colon cancer. So in terms of, transferring microbiomes such as lactose-free. But we are looking at other radiation damage, that doesn't involve the colon.
Thank you. Nice work. Thank you again.
Thank you. Thank you.
Well, I think, Jenny, you see, you got too many questions. I feel bad, actually. I think we're going to just have a last question. And if anyone have a question, please send Jenny email. I bet she's happy to answer any question. So Alain, you want to go ahead and ask question? You're the last one. Okay, I'm gonna just read his question. Thank you very much. I have a question about NRP12. Is there any typical inflammasome activating pathway of NRP12?
Yeah.
Except I think
You're
it's
saying, yeah. So that's the study done from Leanne and Fitzgerald, and I don't know of any, but I don't discount that study either, because in several cases, several inflammasome proteins have the same functions as NRP12. So a typical example is NRP6. So NRP6 is defined as an inflammasome. It actually has specific ligands, like I was talking about, right? So single strand, so RNA, also lipoteichoic acid. There are several activators of NRP6, but there's a whole host of papers showing that it negatively regulates NF-kappa B or ERK pathways and downregulates inflammatory cytokines. In NRP12, Yersinia is the one case I know about. In all the other studies that I've seen, it seems like inflammation is increased by the absence of NRP12. But like I say, I don't discount that as an inflammasome because there is that study. So it could be that we just haven't found the right stimulus.
Okay, so thank you so much, Jenny. I mean,
Thank you so much, Tron, for
it's a great talk. There's a lot of great questions. I mean, thank you so much for staying with us for so long time. Hope I can see you in person soon and stay safe. So thank you everyone for participating in today's seminar, and then hopefully I see you guys next week. Thank you so much, Jenny.
Great to see everybody.
Yeah, good night.
Okay, stay safe. Bye.
Yeah, bye-bye.
Bye-bye.